Okay, time to start. It's a pleasure today to introduce a new chum to this building. Hasn't been seen for several days. John Rasko is a medical graduate from the University of Sydney. 20 years ago, he came to Melbourne and joined the Cancer Research Unit as a PhD student. After that, he went to Seattle, to the Fred Hutchinson Centre, and then following that, he returned to Sydney, to Royal Prince Alfred Hospital and the Centennial Institute. He has a number of titles at Prince Alfred, which I can't remember without looking at my list, but it sounds pretty important. Head of Cell and Molecular Therapies. Imagine that. And he's got a corresponding title in the Centennial Institute. Now, it's a great pleasure to have you back, John. What he's talking about today has just appeared in Cell, and I advise you to read it. It's quite complicated, and I advise you to pay attention. If what he's about to tell us is verifiable, if it's more generally applicable to other tissues, and particularly if what he's describing is not just something that goes along with maturation, but is what mediates maturation, then I think you'll be listening to probably one of the most important seminars that you'll hear in this decade. So pay attention and see what you think.
You made me cry, Professor Metcalf. I feel like I'm at home. 20 years, long time. Only happy memories of this place. Two little babies we had here. Don had nothing to do with that, I don't think, but we haven't had the genotyping done at this stage. But you know, both of them now at university, and it all started here. And so many friends in the audience, and it's a joy to be back, even though I've never heard Don bullshit so much about the significance of my work. Nevertheless, I will try and convince you that there's something interesting happening in this area. So today I have the task of giving you a little bit of background as to an excuse as to why I'm interested in this area, and spend about a third of my talk on a bit of background, how I'm in the field, and then really focusing on this concept of intron retention, which I'd like to try and convince you of may be an important component of one of the multiple layers that so many of you in this audience work on in terms of determining gene expression. And so starting from the most simple, because I'm a simple person, background, we are all familiar with the paradigm, DNA goes to RNA to protein, and we're all familiar with what I'll call a series of decorations in these pathways that many of us have worked on, either directly or indirectly over our careers, as listed here. And I guess the focus of the next 50 minutes or so will be to try and convince you that non-coding RNAs play a role. And this is something that's been taken up and championed by none less than John Maddock throughout his career, but many others as well. And I guess for about a decade, I've been interested in this field myself as part of an interest in hemopoiesis, which originated back in this place and even before that. And just one indulgence, I do remember very vividly as an enthusiastic medical student, but also then as a trainee in the hospital at Royal Prince Alfred in Sydney, coming down to Weehigh probably in my third or fourth year as a resident, and asking Don whether he might take me on as a PhD student, and he said, well, look, it's possible, I need to check things out and make sure that everything's okay, but it sounds like that might be possible. And I said, but Professor, I have at least 2 or 3 years more to go in my training to do the College of Pathology, College of Physician, yada yada, and you know, I'm pretty much committed to that. And he said, stop wasting your time with all that crap and get down here. And I was the only time I ignored the great professor and disobeyed him, but never again. In any case, what we're going to be talking about is subtypes of RNA for part of the majority of today's talk. So RNA, of course, is a functional molecule. We completely recognise the obvious parts of the coding and mRNAs, but in particular we're going to be focusing on those components which are non-coding RNAs, familiar to all of you, divided into the transcriptional RNAs, and many people interested in the long non-coding RNAs, which I won't be talking on today, but what I will be speaking about is the small non-coding RNAs, which are divided up into many different flavours as listed here, and I guess one component of that small RNA field that I haven't, that I'd like to just mention briefly is some years ago with our local bioinformatician William Ritchie, who really for the last 5 years in close collaboration within my program and is now head of bioinformatics at Centenary, has basically followed my interest into the myelopoiesis field, and we just simply asked a question when we had some deep sequencing data in myeloid cells to basically allocate small RNA species to different classes as listed here. And even many years ago when we started this work, by far the majority of mRNAs we couldn't classify based on this simple classification at the time that we did it. And that actually led to a productive collaboration with John Maddock and Ryan Taft, which identified arguably a new class of small non-coding RNAs called the SpliRNAs, which are demonstrably shorter than microRNAs, which nevertheless have a striking map to 3' ends of spliced donor site and exons, and clearly developmentally regulated and clearly involved, or at least functionally involved by association with aspects of modifications of transcription. And I guess the only data that I wanted to show is, because I'll be talking about microRNAs for at least the next 5 or 10 minutes, is that these SpliRNAs that were published a few years ago now, not only did they show this extraordinary mapping to splice sites, but also clearly their biogenesis is separate to that of microRNAs, because the microRNA biogenesis pathway is completely dependent on DICER, and in this case in the DGCR8 knockouts and the DICER knockouts, you can clearly see the same presence of these SpliRNAs being lined up in the same way in these knocked out cells. So another class of RNAs that are clearly being manufactured, at least within the cell, by a pathway that's completely separate to the microRNA class, which so many people are interested in these days. I first really got transfigured in the small non-coding RNA field by this experiment. I thought this was a shocker. I'd always been interested, since even before coming here, in oncogenes. It was the heady days of Bob Weinberg and Mick and Suzanne and Jerry's extraordinary work. And here was a paper that was using Suzanne and Jerry's emu-mic mice and basically knocking in what now famously is the 1719B polycystron of microRNAs. So only small non-coding RNAs. I wrote an editorial once saying, throwing the baby out with the bathwater, and I was only saying to Davo earlier today, you know, I'd take my northerns to Paul Warren, who'd be suffering kind of and shaking his head going, you don't know how to do a northern if your life depended on it, Rasko. And I said, what are those bands at the bottom of the northern? Well, just let them run out, throw out the baby with the bathwater. These were the small non-coding RNAs that we used to ignore as recently as 15 years ago when we ran our northerns. But here was knocking in just a group of microRNAs, just a handful of them, and dramatically accelerating oncogenesis in the emu-mic mice. And that's, you know, almost now 10 years ago. And that really indicated to me that these genes are important. And I guess that's not news to anybody now in this room, but I think that was a seminal science paper back in 2005. Now we know that microRNAs are compellingly involved in many aspects of neoplasia. You can go through the evidence listed here, but the so-called oncomeres, the transforming microRNAs, half of the mapped microRNAs are located at fragile sites and genomic regions involved in cancer. We know that the MIR142 precursor is involved in the translocation of the 817. You can see the list. The LET7 is one of the most famous ones, the Nobel Prize winning, if you like, microRNA at the start is underexpressed in lung cancer, both in human and in cell lines. The polycystron I've mentioned before in acute myeloid leukemia and others, as well as the Burkitt lymphoma 155. And I guess that has led to profiles and signatures that have made significant indications that these are changing biology in cancers. This is the data from chronic lymphocytic leukemia in the New England Journal. This is the data from acute myeloid leukemia, purely stratified on microRNA signatures as shown here. I mentioned before biogenesis, and this is just a slide to remind those of you who aren't familiar that the biochemistry of microRNA production is very well defined, DGCR8 and DROSHA being those enzymes which convert the microRNA, the primary transcript from a microRNA gene, into a pre-mRNA, which then it gets exported out of the nucleus and processed through the typical dicer-1 mediated splicing or cutting, which then allows that to be incorporated into the RNA-induced silencing complex, and then mediating several different mechanisms by which gene expression is modulated and a number of different targets, some would say in the thousands, for each single microRNA, and they are listed here. One is by cleavage of target mRNAs, typically like an siRNA mediated cleavage. Another is by translational repression, and then, of course, the deadenylation mechanism as listed here. So I just wanted to give a background as to how I got involved in this. And when the field of small RNAs was starting to be discussed, and I guess a person who became very important to me in this field was David Martin, who was working at the Victor Chang, but I came to know him very well at the Hutch in Seattle during my postdoctoral years, 96 to 99, and he was very, very interested in gene expression and said, you know, small RNAs are the next big thing. And when I've seen Simple Waves started thinking about that, probably in about 2002, 2003, we didn't even have a means of quantifying these small RNAs. All we could do was run pretty crappy northerns. There weren't flash page extractions or anything. And so I figured that it might be worthwhile trying to work out a mechanism by which we could amplify microRNAs and quantify them using what was very familiar to me, which RT-PCR technology. And so we tried every trick in the book, rolling circle amplification, PCR, and in the end we used a technology that we reported in a now obscure journal, but it was the inaugural issue, and I was terribly impressed with myself doing that at the time. But very rapidly this technology became unnecessary, or at least superfluous, because so many companies jumped on the bandwagon and replicated or indeed were in parallel developing similar technologies, which all required, the trick was to basically create a fusion with the RNA and amplify from either a 3' or a 5' extension of the microRNA itself, which being 22 nucleotides couldn't actually generate a product by itself. And so that allowed us then to adopt pretty early on newer technologies, in particular the TACMAN low-density array and most recently the nanostring technologies that allowed us to do a number of studies to define the extent of expression and then correlate that functionally in various diseases. And I'll just give you one snapshot which has led to some substantial collaborations and some work that now is proceeding very well. And the example that I've chosen today to highlight is microRNAs in chronic myeloid leukemia. And so what we set out in about 2007 to do was to take newly diagnosed chronic myeloid leukemia patients in collaboration with Ali Turhan in Paris at the time, and then take their peripheral blood prior to the tyrosine kinase inhibitor imatinib or Glyvec, and then basically take their blood at days 1, 7 and 14, apply TACMAN low-density array technology and validate those microRNAs that were identified as being up or down in populations, and then see what we could work out in terms of correlations. And so we were able to generate quite a bit of data, and this is just a snapshot of some of the microRNAs that we were able to show, and we were able to discern patterns. So here's two examples of microRNAs that were generally up and two examples of microRNAs that were generally down in the majority of the patients, and those particular examples were then extended to consider their targets and then look at gene ontology, for example, and work out signatures which were then validated in another reference group that we were able to do prospectively. And the conclusion from that relatively simple study was that, again, microRNAs could be used to correlate clinical outcome, in this case correlating with the SOCAL score, which is a prognostic indicator, that they could be used as novel biomarkers, which we are now using actually in prospective studies to predict which particular tyrosine kinase inhibitors might be useful, or indeed those patients who might be resistant to tyrosine kinase inhibition. And that's a project that's currently funded and underway with Tim Hughes and Deb White down in Adelaide, who are really leaders in the field of clinical use of tyrosine kinase inhibition and monitoring. Charles Mulligan's name also in that, and a PhD student in my lab, Jane Gordon, who's a haematologist. Ultimately, the trajectory of the microRNA stuff is obviously to explore the possibility of therapeutic strategies. And I guess recently, this is one of a number of reviews variously published, but there are obviously two ways that you can imagine that we might be able to go about using our knowledge of microRNAs as therapeutics. This is in a relatively advanced phase. In particular, Phase 3 and beyond studies now are pursuing hepatitis C as a target for small RNA therapeutics. But in the case of diseases like cancer, leukemia, myeloma and lymphoma, the idea would be that there are two strands by which there can be perturbations that might be targetable using small RNA molecules, and they're listed here. The loss of a tumour suppressor gene, which then could lead to a therapeutic approach by which we then either recover expression of that tumour suppressor by the mechanisms listed here, or the converse, of course, the activation of an oncogene which could then be inhibited by interfering with the expression of that particular oncogene using microRNA mimics, or indeed a gene therapy-based approach, which has been a hobby of mine for as long as I can remember. So what I wanted to wind up this introductory phase of the talk is really where we focused most of our attention, and most of our publications over the last decade in the small RNA field have been more focused on target prediction and working out the informatics of what it is that microRNAs are doing in cells on a specific basis. And so that has been a bioinformatics focus predominantly. And as you would know, for those of you interested in understanding what the targets of these 22 nucleotide long non-coding microRNAs are, really bioinformatics has offered us vast amounts of data. Every microRNA, potentially, depending on whatever calculation or algorithm you use, will predict hundreds, if not thousands, of potential targets. But the confirmation is laborious, tedious, and time-consuming, and often not compelling. So most of the informatics technologies that are used, the algorithms that many of you will have visited on the web, things like TargetScan and Miranda, are based on free energy calculations, predicting how well a microRNA will bind with a given cognate target. Our interest has been in a complementary field of anti-correlation, which I'll devote a slide to in a second. And of course, gene ontology is another tool that many people use to try and, I guess, bring together data based on the idea that microRNAs target specific mRNAs which have overall common functionalities. The 3' UTR reporter system, I still think and have argued that this is the gold standard, namely both providing a 3' target sequence within a UTR and mutating that target sequence to demonstrate that it is indeed the target of a given microRNA. I still think is the gold standard that I require to be convinced that a given microRNA and a given target 3' UTR are paired. But nowadays, the cross-linking immunoprecipitation offers a global approach to identifying the possibility of targets. So let me just indulge one slide talking about how we went about thinking about predicting targets based on the knowledge that there were a lot of algorithms available that use free energy calculations. And one of the first things that we did was simply to ask the question, what are the commonalities between predictions between the different algorithms that are available, all based on just simple free energy calculations. And the thing that shocked William Ritchie and myself when we were first exploring this was the simple fact that the overlap was shockingly rare. And I'm certain everybody in this audience who've sought to find an interesting microRNA and then try and understand its targets, you sit down and you go and you look up your various algorithms, you look up Miranda, you look up TargetScan, and then what invariably happens in the literature is people look at the commonality, the Venn diagram of which of the targets are common to the two algorithms, which are both using the same free energy calculations for all intents and purposes. And the commonality is rare. And what this is actually to highlight the fact that it even depends on which 3' UTR library the particular algorithm is accessing, because the overlap of the libraries is not even 100%. They're not even using a commonality of denominators, if you will. And so here we are basically combining the two denominators, and even then the overlap is not 100%. You would think that if you're just calculating a free energy, it's a mathematical calculation, you would find a near 100% commonality, but never reached 50%. And so that was something that we wanted to raise as an issue, but we've taken that considerably further now by providing our own algorithm, which is the mimi-RNA algorithm, which has been around now for a few years. And this is a snapshot of the website, which basically turns the whole thing on its head. And I used to kind of arrogantly say, well, I can predict the target of a microRNA without knowing its sequence, which was provocative and designed to be so. But that was the whole point of this is not based on any free energy calculations. It's based on simply the idea that when microRNAs target a given mRNA by direct targeting and degradation, they are going to naturally, if you take vast data sets of different cell lines and have all of the microRNA data and all of the mRNA data in those given cell lines, you can anti-correlate microRNAs and the mRNAs. And if you find in many different instances statistically significant anti-correlation, so where a microRNA is up and an mRNA is consistently down and vice versa, then it's possible that that is a true cognate target and microRNA pair. And so that's exactly what mimi-RNA does. It's an expandable database which we can consistently giving more data to. And it was named after the Aboriginal Mimi spirits because I thought that they look a little bit like the hairpin of a microRNA in its biogenesis. And so mimi-RNA is the tip of our hat to the Aboriginal Mimi spirits, but also another way of generating predicted targets globally and indeed individually. And for those of you interested in predicting targets, you can go down and select any one of the tasks that you want to undertake and decide which way to go. So I will move on then to what I want to spend most of my time on, which is, as Don generously said, perhaps a new way of thinking about another layer of gene expression control, which is intron retention. And so I guess I've laid the foundation for saying why I'm interested in gene expression and why I'm interested in RNA controls. But ever since I was here, my compulsion has been to explore that in model systems which were tractable and well-defined. And mouse granular poiesis for me, even though I would claim still not to be anything like an expert, is an area that I find extremely well-defined and extremely tractable. And so everyone in this audience recognises this as the terminal phases of mouse granulopoiesis. The reasons why it is so beautiful is that we know that there are distinct phases of transcriptional and translational changes in those three phases. The exquisite and still beautiful changes in the nuclear morphology that we all witness when we're looking at these cells under the microscope, and the very fact that in the mouse we get these gorgeous circular donut-shaped nuclei. In humans, the donut is not closed, but they are very, very similar. And I guess as an underlying theme, as I was thinking about this project over the last 4 years now, we have been directed towards thinking about a very fundamental question that one never really has the time or inclination to think about, which is why it is that the granulocyte nucleus is folded such and what function that might confer. But the beauty of it and the conservation of the shape of the granulocyte nucleus is unique. And so in the hand-waving way back in medical school, so the cells can squeeze out through the endothelium and get into the pus-filled, bacteria-filled boils that they need to get into. Well, it just doesn't make any sense whatsoever. That's not the reason, surely, because other cells can do similar kind of things, not without having such an obscurely shaped nucleus that is unique to this lineage. So let's move on then to the focus of what I'll be talking about, which is alternative splicing, essentially. Pretty much every gene in the genome is subject to alternative splicing, certainly 95% and upwards. And the ostensive reason that we have is to generate protein diversity through putting in or excluding cassettes of exons as listed here. And those familiar alternative splicing mechanisms as listed to alternate the 5' splice site, the 3' splice site, or to exclude an exon, thereby including or excluding cassettes of peptides which make up those which encode those exonic sequences, those peptide sequences. But the one odd man out, and the one that we'll be speaking about today, is the concept of intron retention, which is poorly understood, I think it would be fair to say, because predominantly most people think that it's just noise. Back in the good old days, of course, when people were shocked by the fact that introns existed, they were faced with the problem of producing recombinant proteins and needed to use the trick, of course, of reverse transcription to exclude the introns in order to have cDNAs. Most people don't even give any thought to that. But clearly, the reason for most people wanting to exclude introns as an area of interest is because we all know that most introns have in all three frames, stop codons which induce premature termination complex formation and nonsense-mediated decay. And so intron retention has long been regarded as a mechanism of monitoring the genome or indeed protecting the cell from the possibility of producing aberrant peptides which might have a dominant negative effect or a detrimental effect on the cell. So introns giving rise to a mature pre-mRNA then are included in the processing outside of the nucleus of the mRNA, and that is what we define as intron retention. So I'll be speaking about IR or intron retention throughout the rest of the talk. What I define as intron retention is a mature mRNA, a poly-A plus mRNA that's found its way into the cytoplasm following all of the processing and that has still got an intron that is retained. It often is thought to occur following missplicing, and we all know that there are diseases, both somatic and germline, which can affect splicing and lead to the aberrant retention of introns, which then lead to abnormal protein production and either a truncated protein or a degraded protein, which leads to a disease state. So this possibility of dominant negative proteins can potentially be harmful, and that's at least anthropomorphically, why we have a mechanism that is so well evolved called nonsense-mediated decay, which leads to the degradation of anything that retains an intron that has premature stop codon detected by a mechanism that parses sequences during the pioneering round of translation. And if a premature stop codon a certain distance away from the poly-A sequence is detected, then that sequence is marked for degradation by a complex biochemistry known as nonsense-mediated decay. We know that the phenomenon of nonsense-mediated decay is essential to hemopoiesis, at least at the early stages of the stem and progenitor phases, because a friend and colleague of ours, Bo Posse, has published a very beautiful work in mice showing really a partial knockout, to be frank, but certainly a near knockout, showing that NMD in this G&D paper is essential for hematopoietic stem and progenitor cells because it is essential for eliminating DNA, programmed DNA rearrangements. So here's our experiment. About 4.5 years ago, we decided to dissect the process of promyelocytes to myelocytes to granulocytes by performing the full omics that we could possibly do, by which I mean mRNA-seq to the depth of about 60 million reads in all of these phases, small RNA omics, microRNAs, proteomics as best we could, and then, of course, complementing that with informatics. And the informatics was absolutely key. If William Ritchie hadn't been there, we wouldn't have been able to do the experiment that I'm about to describe, and that is because we were particularly interested, at least for the purposes of this talk, in this phenomenon of intron retention. And what I'm talking about now here is not the pathological state of a splice site mutation where an intron is retained, and that's an aberrant pathological phenomenon. I'm talking about looking at exclusively normal white blood cells in normal mice and in normal humans. And the problem is that introns, being what they are low complexity. They are typically sequences that are not as highly conserved as exonic sequences. Indeed, that's one way that Terry over many years will have identified exonic sequences compared to intronic sequences because of their high conservation rates with encoding sequences. But the very fact that they are low complexity means that most of us in this room take for granted the deep sequencing data that we get from our Illuminas or our solids, because there's a trick that nobody really talks about very much outside of now this area, which is that we mask the sequences that we receive. And that's bioinformatics talk for saying it's too difficult. I can't map those sequences to anything. I can't map those sequences to a unique location in the genome, and so rather than kind of even do anything with them, I'm going to throw them out. So when you get your sequencing data, typically you get a masked sequence that has already been mapped to the genome, and anything that couldn't be uniquely mapped has been thrown out. And as a consequence, a vast amount of data, it's not trivial, it's a vast amount of data, of sequencing data, is thrown away. We wanted to retrieve that, but in the most rigorous way we possibly could, in order to see whether or not we could detect low complexity sequences in introns that might otherwise have been ignored. Because people ask me, so John, you're trying to convince me that you've discovered something new. Lots of people have been doing sequencing a lot longer than you have, and they haven't seen this. In fact, in retrospect, a lot of people have seen this and ignored it, but we've tuned it up in a sense by using a bioinformatics tool. And I'll just spend a moment mentioning this because this was really key. William wrote code that he's called IR Finder, or Intron Retention Finder, not very imaginatively, which allowed him to basically look at the sequences that we had, the sequencing that we had available, and basically make certain that every intron that we were about to consider had overlapped sequences between exon-intron boundaries. And then, basically, we wanted to make sure that the intronic sequence for a given intron was at least 50% uniquely mapped. So that was a minimum criterion in the first instance. But then we went back to the unmasked data set and said, can we fill in the remainder of those previously unmapped areas? Can we fill that in with sequences that might have otherwise been thrown out by virtue of the masking process? And at a minimum, we required that we cover it 90% in its totality with the retrieved sequences. And then if so, we were then able to go back and calculate what the intron retention level is. So this was a means of identifying, putting a number on the retention of introns, and that's what Intron Finder does. The other point that I want to make, and it's just as important, is because we had 3 sequential steps in maturation and differentiation, we were able to at least distinguish noise from real differences. And I'll highlight that in the data that I should just show in a second. But because we had sequential stages, we weren't just looking at one snapshot, we were able to see changes. And those dynamic changes were extremely important in allowing us to be more and more confident several years ago that we were onto something new. One of the first questions that we needed to consider was what proportion of the transcripts that we were seeing were exhibiting intron retention. I'm going to describe that we had 86 genes, and that's what's in the paper, that gave evidence of intron retention at more than 5% of the genes that we identified. And that's true, but we believe that Intron Retention Finder and the intron retention detection technologies that we developed are actually grossly underestimating intron retention. We think that we were extremely rigorous and stringent in the way that we applied these algorithms, but we actually think that there's probably a vastly larger number of genes that are exhibiting some amount of intron retention during the processing of their mature mRNAs. But this is basically a list of the 86 genes here in promyelocytes, myelocytes, and granulocytes, asking the question, what is the percentage of intron retaining sequence out of the totality of sequences that we got in our mRNA-seq data? And basically what you can see is that in some of the genes, there is a substantial increase of intron retention as the cells differentiate from promyelocytes to granulocytes. So an overall acquisition of the retention of introns as these cells mature. But in order to convince you that, and importantly, I guess some of the genes had 95% of their transcripts as intron-retaining transcripts. Other ones, 25%, 50%, and as low as 5%, which is where our cutoff occurred. This is to convince you of the raw data. So this is IG Viewer, probably familiar to many of you, and this is 6 different genes showing evidence of intron retention out of the 86. But to make it easier, I'm just going to show you the one gene that will become interesting later on in the talk as well, which is a very, very good example. It's one of the examples of the genes which had one of the very highest levels of intron retention, and this is Lamin B1. the nuclear membrane gene. And here you've got promyelocytes in the top reads, myelocytes in the middle, and granulocytes here. This is the gene. Here is the transcript start site. The little blips, of course, are exonic sequences, and in between are the intervening intronic sequences. And these are the reads. These are mouse data, and this is human data for just mature granulocytes shown here, and the very, very similar human sequence, the human gene sequence as shown here on the blue line there for human. And so just focusing on these reads here, and the scale is the same on each of these three here, and so on, you can see that the right-hand side of the gene is basically acquiring the retention of reads in the intronic sequences as granular poiesis is maturing. And in this red bar here, we're just simply selecting one location. We did multiple windows throughout many of the introns, but this is basically giving you a sense here in the Intron Retention finder numbers of normalized intronic expression from promyelocytes, which are defined as 1, to myelocytes and granulocytes. And what you can see here is a 3,000-fold increase in the retention of introns as we go from promyelocytes to granulocytes, and similar kind of numbers observed in granulocytes, suggesting to us that there was a retention of these introns, but importantly, a specific retention of introns towards the right-hand side of the gene of the transcript as shown here. So none of these introns here were retained at any point during differentiation. And I can go back to this later on. It may be some component of an artifact because all of these reads were poly A+. So it may be that we had 3' bias to some extent, but or it could be real. The important point was that there was a differential acquisition of specific introns within a particular gene, and indeed this is the case for 86 other genes, as I mentioned earlier. So so I should also say that the correlation of human and mouse intron retention was very, very striking, and also led us to believe that this may be a conserved mechanism in granulopoiesis, and something we'll be exploring a little bit further as we go on. Because we had proteomic data, we were also able to ask the question, were those genes that were affected specifically by intron retention also exhibiting a loss of protein expression? And the answer was yes. We had mass spectrometry data for 1,500 proteins or more, and the presence of intron retention correlated strongly with decreased protein expression in granulocytes compared to promyelocytes. And that's just a snapshot of some of the data that we showed in the table. Before I go on, I have to deal with an extremely trivial explanation for the data that I presented so far, and that is that all we're doing is reading unprocessed mRNAs, mRNAs that happen to be in the nucleus, and it hasn't had time to splice out the intron yet, and so this whole story collapses in a house of cards. And so what we needed to show very early on is that not only is this poly A+ mRNA, but also that it's processed mRNA, and evidence of the processing, of course, is that it's been put into the cytoplasm. And so here's our positive nuclear enrichment here on the left. Here's our positive cytoplasmic enrichment shown on the right. And here are all of the genes, showing that all of the genes that we identified high-level intron retention in are enriched in the cytoplasm, suggesting again that these are mature, fully processed poly A+ mRNAs that have failed to splice out their introns during normal granulopoiesis. Okay, so and as I mentioned, these peptides do not contain any coding peptides that we could ever detect. Sorry, I didn't mention that. In other words, the intronic sequences themselves we did not detect as small peptide sequences. There wasn't any magical little subgenes within introns that were coding for peptides that we might have otherwise missed. So what are these genes? Could we discern any patterns when we looked at what types of genes we were detecting that were exhibiting intron retention? And so this is a gene ontology heat map that I'm sure many of you are familiar with, separating out those genes which were exhibiting any form of splice isoform expression, any form of exonic inclusion or exclusion or different use of 5', 3' intron exon boundaries, versus those genes that exhibited exclusively intron retention. And what you can see is when you look at all of the genes, which is the majority of genes that exhibited splice isoform expression, some of the genes were low in promyelocytes going up, some of the genes were high going down, some of the genes were higgledy-piggledy higher in the middle. But when we looked at intron retention, for by far the majority of genes, and this 10% or 15% here may be slightly aberrant in as much as we didn't look formally, although we've had a peek at cells even earlier than promyelocytes. So these may simply reflect that there are cells that even have lower levels of intron retention earlier. But by far the majority of the genes acquired intron retention as granulopoiesis was proceeding, in distinction to the majority of splice isoforms, which are up and down variously depending on what particular gene is being expressed at that particular phase of differentiation. And further, when we actually asked the question of gene ontology, we found that even though these genes, as you might expect, because they're the majority, were all correlated with leukocyte and hemopoiesis, when we specifically looked at intron-retaining genes, we found a striking enrichment for the nuclear lamina and the nuclear periphery. So unlike the majority of other genes susceptible, the genes were expressed, or the genes that were alternatively spliced, we found that the genes which were alternatively retaining introns were enriched for genes that were specific to the nuclear lamina and the nuclear periphery. We also wanted to ask specifically if there were any features of these introns that were being retained, and what we found also compellingly in mouse data and in human granulocyte data was that there was a striking, statistically significantly increased GC content percentage in the introns that were retained as opposed to the introns that were not retained, suggesting that there may be intron intrinsic determinants that allow those particular introns to be retained. And that's something that we hope to explore a little bit further, but that's the earliest sense that there may be intron intrinsic factors involved in determining intron retention. But why are introns being retained at all? Well, we could ask that question by looking back at our mRNA-seq data and looking at our proteomics data, but this time not looking at intron retention, but looking at the level of gene expression in mRNAs and in proteins for splice factors that determine either exon definition, as shown here in this left part, or lariat formation and cleavage, shown in this right part. And this is a list of all of the genes that we had data on for both mRNA, shown in the squares, and proteomics, shown in the circles. And what the general impression is that there is a large number of genes in the exon-defining phase of splicing that are down-regulated in granulocytes compared to promyelocytes. So they're at a certain level in promyelocytes, but as differentiation proceeds, these splicing factors that are involved in exon definition are down-regulated. In contrast, the remaining part of the splicing machinery, which is involved in lariat formation and cleavage, stayed relatively stable. So a nice kind of internal control, if you will. But again, suggesting that maybe intron retention is associated with differential expression of splicing factors. And I guess for those of you who are not familiar, certainly in the hematology world and even in the oncology world, there's been extraordinary excitement over the last few years about recognising mutations in splicing factors due in diseases like myelodysplasias and other malignant conditions, which have been quite shocking, because anybody with any kind of sense for decades would have told you that it's impossible to imagine that there are heterozygote mutations in splicing factors, let alone double mutations in splicing factors, because any man and his dog will tell you that splicing factors are so absolutely intrinsically important and essential to the production of genes generally that any aberrancy in splicing factors would lead to the cell to just die and collapse. And so this was a shocking discovery that was made only in the last few years, that human malignancies have got very clearly refractory anemia with excess blasts, and so 85% of those myelodysplasias have mutations in one of these splicing factors listed here, SF3B. So clearly, we think that this may give us a hint that this may be relevant also not only to normal myelopoiesis, but potentially of relevance to malignant myelopoiesis as well. So for the remaining, not for the remaining part, but for the next little while, I'll be speaking about Lamin B1, which was the gene with one of the highest levels of intron retention that we detected. And so Lamin B1 is part of the nuclear lamina. It's one of the lamin proteins, as listed here in this lovely review that was published, and is known to be important in maintaining nuclear stability in architecture. And if you'll bear with me, I guess what I'm trying to do is tie together an observation that I planted the seed of earlier today, which was the shape of granulocyte nuclei and the possibility of how this might tie in with this interesting aspect, and also the gene ontology data that I showed earlier, talking about the nuclear periphery and the nuclear lamina. But before I do, I want to just simply give you a little bit more detail, and using Lamin B as an example, which was one of a number of examples, showing that how beautiful intron retention is specific for in different genes, and so we're going to use Lamin B1 as an example. So this upper panel here is basically showing the expression, and I don't know how well you can see it, but exons on the top and introns on the bottom. So this is the way the gene is structured. And basically we're looking at promyelocytes, myelocytes and granulocytes gene expression level by qRT-PCR. And what you can see is the exons are going down. The overall gene expression of Lamin B1 is going down during maturation of granulopoiesis. But the retention of introns, in particular the 3' introns, is dramatic. This is a log scale, dramatically going up. There are many, many more intronic reads as differentiation proceeds. And these are M-pro cells over a time course differentiating from what are effectively promyelocytes towards predominantly granulocytes, showing predominantly the same data. And of course, I have to show you the Western blot and the immunofluorescence to confirm that indeed, yes, gene expression is going down because exonic sequences are going down, even though intronic sequences are going up. Clearly, these are the retained intronic sequences that we were able to capture because of the depth of the sequencing that we have available. But here's the protein data. Clearly, Lamin B is going down, as everybody knows, and is well published. There's no news there. And in immunofluorescence, you can't see Lamin B1 expressed on the nuclear laminin, the lamina of the nucleus in granulocytes, but clearly there it is in promyelocytes. So now we have to ask the question, are these intron retaining transcripts truly being degraded by nonsense-mediated decay? Are they activating the pathways that I introduced earlier? And so what we needed to do to answer that question was to block nonsense-mediated decay using a couple of different methodologies that were available to us, and taking advantage of the fact that the biochemistry of nonsense-mediated decay has been very well dissected. And what we know is that this particular component, SMG1, which phosphorylates UPF1, is blocked, believe it or not, by caffeine, that happy drug that many of us are addicted to, particularly Dr. Lindemann, who I know has had trouble with. But anyway, this aspect of nonsense-mediated decay, which can be blocked and has been for many, many years blocked using caffeine, which is known to have pleiotropic effects, but nonetheless is relatively well accepted as a specific way of knocking down nonsense-mediated decay. And so in order to use the best technology that we had available, we used the current one published in Oncogene 2007. We took the primary sorted myeloid cells that you're now familiar with, that we've been dealing with. We exposed them to a short caffeine NMD inhibition assay in the presence of actin and myosin D to block any other factors that we might be distracted with, and then basically quantify the retention of specific aspects of this particular gene. And what you can see, I think this is really compelling and beautiful data. What you can see in promyelocytes compared to myelocytes compared to granulocytes, so black, red, blue, that clearly the exonic sequences are going between the two are unchanged, whether caffeine is present or not, but there's a general trend downwards, again, on this scale, on this log scale. But the introns are being, if you only look at the non-caffeine treated, you see, yes, there are more introns being acquired, as I've shown you in previous slides, but when you put caffeine there to block the degradation by NMD, you see a massive revelation of intron-retaining reads by qRT-PCR present in the two adjoining introns, suggesting that NMD is degrading these transcripts, and because by blocking it, we're seeing a lot more of those transcripts that are retaining introns, and the exact same data can be demonstrated in the differentiating MPRO model, in vitro model of this cell line that I showed earlier. Again, exonic sequences relatively maintained or degraded, but introns dramatically increased after NMD inhibition using caffeine. Using an entirely different methodology to block nonsense-mediated decay, we used siRNA of UPF1 to reduce NMD in a more specific way, because caffeine has its detractors. And essentially the data is exactly the same. Here again now, we're showing the entirety of the exonic sequences of different, different exonic sequences and adjacent intronic sequences in different genes. Here's Lamin B1, here's a bunch of other genes, all of which exhibited intron retention, and again the same data by inhibiting UPF1 by siRNA and blocking NMD, we show retention, even a greater level of intronic sequences. We know that I felt that this was sufficiently compelling to suggest that intron retention was occurring, but one of our reviewers was quite adamant that he couldn't believe that this was the case unless we convinced him that this wasn't also somehow linked to transcription. My feeling was that this is a new layer of gene expression control, and it could be the case that transcription goes up in a gene, you still get intron retention. Transcription goes down a gene, you still get intron retention. It didn't make any difference to me. But one of the reviewers said, I don't believe you. I think you need to show to us that transcription isn't going down before I believe that this is a real general effect on gene expression. And so myself reluctantly, but the postdocs who were keen on publishing this in obscure journals were keen to do this experiment, so we used the Invitrogen click-it technology to look exclusively at transcription, early transcription, so in other words, transcription initiation. Basically, these are nascent transcripts. And so these are the very well-described down-regulated genes, in fact, both of these are positive controls, if you like. This is showing that we could detect nascent downregulation of gene transcription during differentiation and nascent upregulation of gene transcription during differentiation. And so now we can ask the question of all of the intron-retained genes, what is the change in gene level of expression, knowing that we can detect things with quite a substantial level of sensitivity. Again, a log scale shown here. And here are the changes in by far the majority of the intron-retaining genes. Most of them either stay completely stable during myelopoiesis or even go up a little bit. So the nascent transcripts is completely independent of the overall degradation in expression of genes that occurred in all of these, and very much the shutdown of protein expression that occurred in the majority of these genes as well. The last part of the talk will be just to give one example in vivo of perturbing this system, which I felt was important, even though not necessarily as compelling as the previous data. And so what we wanted to do was to prevent a cell from excising introns in a particular example. And because we'd spent time looking at Lamin B1 particularly, all we did was we took an intronless Lamin B1 gene. I like to use that phrase because it sounds like it's something particularly clever, but it's really the cDNA, because that's what an intronless gene looks like. We basically expressed Lamin B1 fused with a 2A expression system, this is IRES expression system to GFP in a mouse transplant model, and asked the question, first of all, were we getting physiological levels of Lamin B1 now being expressed for the first time in granulocytes. And so this is a mixing study here. Here's normal C57 black 6 promyelocytes with Lamin B immunofluorescence, and here are the gene-modified granulocytes that have been removed from a mouse 6 weeks after the transplant, showing that indeed now for the first time these granulocytes are expressing Lamin B1, and here's the Western to show what I'm trying to show is comparable physiological levels of Lamin B1 compared to normal promyelocytes. And here's the results in the transplant, basically on the full blood count. And what we saw was quite an impressive and statistically significant reduction in the number of granulocytes in animals that have been transduced with an intronless Lamin B1 compared to a control construct that didn't have Lamin B1. And when we looked at these cells, this is DRAC5 staining with a 3D reconstruction, we saw quite a dramatic change. Here's the nice immunofluorescence of these granulocytes showing that Lamin B1 is present compared to the controls which didn't. But the compelling point was that there was a striking change in the nuclear morphology, which I've said I thought was highly reminiscent of a megaloblastic-looking nucleus in a granulocyte. Reminiscent of folate B12 deficiency. And when we enumerated that and quantified it using this kind of technology, we found that there was quite a substantial increase in the nuclear volume. And the impression is that you can see that impression from that particular slide, an increase in the nuclear volume in those cells that were following transplantation. Sorry, I've gone backwards. So, let me summarise. We think that intron retention in granulopoiesis affects a large number of genes. We think that 86 is a conservative estimate. It is the case that this occurs during the right-hand part of myelopoiesis towards granulocytes and leads to reduced protein levels. It's conserved between human and mouse. I showed you the data earlier. It regulates numerous genes involved in nuclear morphology during granulocyte differentiation. It is the case that Lamin B1 and many other genes that exhibited intron retention are degraded by nonsense-mediated decay, so that activation of that automatic mechanism is present. And the persistence of Lamin B1, in other words, perturbing the system of intron retention, reduces granulocyte numbers and increases volume and alters their morphology. So for those of you who like it in a cartoon form, during promyelocytes differentiation towards granulocytes, what we see is a downregulation of specific splicing factors involved in exon definition. These particularly affect high GC content introns, and as a consequence of those factors, or at least at the minimum associated with those changes in those factors, we see an activation of nonsense-mediated decay, degradation of those mRNAs that retain introns, leading to downregulation of expression of those transcripts. So for me, this brings together a lovely multi-layered cake of gene expression, in particular in white blood cells, maybe I would suggest hopefully a previously unrecognised level of gene expression control that involves the alternative splicing machinery, but one that may have well been neglected in normal granulopoiesis up until now. And we believe that there's pretty good evidence that this is true for many other cell differentiation systems. So just offering another level of gene expression control that's possibly been overlooked for reasons that I mentioned earlier due to low complexity reads in intronic sequences. I must spend at least a moment or more thanking the people who were involved in this study. These are the angels who are authors. I should thank Matthias Selbach and Olivia Ebner in Berlin for the work they originally did on P-SILAC and the mass spectrometry data they generated. Also collaborators at the University of New South Wales who did some of the protein bioinformatics, in particular Jason Wong. But really the stars here, William Ritchie in the yellow T-shirt is the bioinformatician, head of bioinformatics at Centenary, and really who drove the IR Finder work and has been absolutely crucial to this work. And Jason Wong, who did the molecular biology. Jeff did the transplants. Leanne Koo, Dadi Gao did helped William with the bioinformatics, a PhD student. Nora Thiong, Maria Gonzalez, and Kincha Bejier at the back, but also Chuck Bailey, who's been with me amazingly for 13 years. My Sandra and Ladina is Chuck Bailey, who's been intrinsic to all of our work. We couldn't do it without substantial funding from various places. And also we had the pleasure of at least having an image generated that tried to summarise in a cartoon manner some aspects of the nucleus, deciding whether to send transcripts towards the garbage can of NMD or whether to make introns excised and producing proteins. I'll stop there and happy to take questions. Thanks very much.
So, time's getting on, but questions, Dave?
Why isn't coffee more toxic?
I think it's dose. It's got to be dose. It is dose.
So how much caffeine would you have to affect cells in a meaningful way?
Jeff has done that experiment. There he is. Jeff Lindemann has done that experiment. Seven double shots a day, was it? Yeah, he's very well done. Huh?
And he's normal?
Well, that's the except, so that's the answer. I don't know. I don't know the answer.
Nick?
So the intron retention you showed is for specific genes and specific introns, it looked like.
Yes, very much.
Avoiding technical artifacts. But the other mechanism you proposed was down-regulation of splicing factors as granulocytes age. Where does specificity come in?
There isn't. I haven't got no answer to your question. I was saying to Ross earlier today that I started this whole project thinking that small RNAs would be the answer. They're not. To alternative splicing, I have no correct answer, Nikos, to what the specificity for particular introns are. And one thing I didn't say that I probably ought is, yes, it is the case that intron retention is conserved between specific genes in mouse and human, but amazingly, it's not necessarily the same introns in the same genes. So the gene is affected by intron retention per se, but it's not necessarily the same intron. So the phenomenon of intron retention initiated NMD is conserved between particular genes, not necessarily the same intron. So even that question is important to consider in the mix of how to work this out. But I have no answer to your absolute, the question we've asked ourselves so many times: why is it that intron and not that intron next door?
Well, upstream of that question, then how do you think it happens that large part of the splicing machinery is downregulated? So do you think that's just, you showed in your mass
I didn't hear what you said, Minnie.
In your mass spec data,
Yeah.
you showed that a lot of the splicing machinery is downregulated.
Yeah.
You think that there is only a few of those factors that are really important, and what's upstream, what is going on?
No idea whatsoever. Yep, sorry. I think, this was a nice functional, an association. It really is not compelling. I mean, let's face it, I think that the nice thing was that we showed exon defining versus there was a difference. That's kind of nice because it suggests there's some specificity. But to Nikos's question and to your question, I have no answer.
Last question, Ben, up the back.
Hey, Ben.
John, is there a link between perturbations in this process and the spliceosome mutations in MDX?
Yeah, I do not know, would love to know. I think there's a lot of deep sequencing data out there, and I think it only takes a little bit of informatics magic to be able to reveal it. I would love to think that this is playing some kind of a role. How specific it would be the question. I mean, is it the fact that the splicing factor mutations are altering particular genes because they're the really important ones? I have no idea again, but I would love to think that this isn't just going to be an obscure mechanism of gene expression control that'll just be lost in antiquity, that it actually does have some relevance to human disease. That's what hopefully is driving us.
On that point, thanks, John. And